Hello, everyone, and thank you for joining us today. I'd like to welcome you all, those of you online and here in person, to today's presentation on the translational research toward conformal treatments for prostate and the brain. A quick note for those of you on the webinar: if you'd like to ask a question of Dr. Lafon, please submit it via the chat function on the upper left portion of the screen at any time during the presentation. We will collect all these questions until the Q&A period begins at the end of the presentation. We will try to get to as many questions as we can during the time we have. And now I'd like to introduce Dr. Cyril Lafon. Dr. Lafon earned his bachelor's degree in physics from the University Blaise Pascal of Clermont-Ferrand, France, and the University of Montreal, Quebec. He received his PhD degree in biomedical engineering from the University of Claude Bernard in Lyon, France, where he developed interstitial HIFU probes in INSERM Unit 281. Dr. Lafon joined the Applied Physics Laboratory of the University of Washington for 2 years as a postdoc fellow. Dr. Lafon worked there on ultrasound-induced hemostasis and development of tissue-mimicking phantoms for HIFU applications. Dr. Lafon was recruited by INSERM Unit 556 in 2002 as a research scientist. His recent research interests focus on modeling ultrasound wave propagation, development of ultrasonic therapeutic devices for thermal ablation or drug delivery, and communication by ultrasound for medical applications. Dr. Lafon.
Thank you for the introduction. So I work for LabTO. LabTO is a laboratory which is part of the University of Lyon and INSERM, which is a French NIH. And TO stands for Therapeutic Application of Ultrasound. So this laboratory is fully dedicated to research of ultrasound therapy. The two keywords of my presentation are translational and conformal, because what we are really doing is trying to bring the device we are developing toward clinical application. And in particular, we try always to develop devices which are conformal to a particular target. So if we look at the timeline of a project, it is very important for us to start from a medical problem. We have a crew of medical doctors that know what are HIFU. So they came to us with an unresolved problem. We discuss together, go through a phase of academic research where we design probes, perform preclinical trials, and then build a device for clinical research that we can evaluate. It's very important for us to work, but an industrial partner follow this process in order to facilitate industrial transfer. So if we look at this 3 phases more into detail, so even if each phase is led by one of the partners, so if we take, for example, the phase 1 with academic development led by the laboratory, it's very important that each actor in the development of the project participated at the different phase. For example, in the phase 1, the doctors must really orient the technical solutions which we decide for, and also the industrial partner must give his opinion in order to make sure that what we are developing is not too complex and will be feasible to reach the market. And same thing for the other phases. For clinical evaluation, it's very important that the academicians are involved in the process of designing the protocols and also looking at the results in order to improve the technology. So by successive iteration, we come up to a solution which is good for the patient and good for the company that will industrialize business. So this takes, of course, many years of research and development. And what I'm going to do during this presentation is illustrate this process to 2 examples. One focused on the design of a device for prostate cancer, and another one on the treatment of brain tumors using opening the blood-brain barrier. So as I said before, it's very important in our strategy to start from a medical problem. Our strategy is always use the ability of ultrasound to be shaped in order to propose a solution which is conformal to the targeted device. So one technical solution for one medical problem. In order to illustrate this, strategy, I've been through a few slides with numerical modeling. I won't be very long. So if we start with a very conventional transducer, it could be used for extracorporeal application. As you can see here, it's a spherical shell, 15 centimeter in diameter and focal length, operating at 1 megahertz. If we shoot with this transducer in tissue with a very normal attenuation, calculate the pressure, the temperature elevation, and the thermal dose using very conventional models. So the Rayleigh integral for the pressure, the Biot transfer equation for the temperature, and the equivalent dose at 43 degrees for the thermal region. So if you want to reach a temperature of 100 degrees at the focus with this transducer, you must use around 100 watts per 5 seconds. So you have a sharp focus, no protocol heating. If you place a layer of fat between the focal distance, between the focus and the transducer, you see this fat layer is very attenuating. So if you run the same simulation, if you want reach also the 100 degrees Celsius at the focus, what you will get is prefocal heating that can be really problematic. So one solution in that case would be to reduce the dimension of the transducer in order to go beyond this attenuative layer. This is exactly what we are doing, for example, with prostate cancer. So I take the transducer with the same F number, so the ratio between the radius of curvature and the focal distance is the same. So now the transducer is smaller, same frequency, so obviously the smaller. And you can see where we get the same focusing. The scale is a bit different, it's why it's larger, but it's the same. If you want to reach this 100 degrees Celsius at the focus, then you need only 25 watts per 5 seconds. And you can improve even more this strategy because you have reduced attenuation, so you can increase the frequency and still get very sharp focus with very efficient heating at the. So this is to say that very important to adjust the shape of the transducer according to the target. If you have a fat layer that prevents the tissue to be treated, then just go beyond it. And this is our vision of the HIFU in general, a therapeutic application of HIFU. We can really make an analogy with the ultrasound scanners. These scanners, they have a common platform with different probes and different acoustic sequences. We will do exactly the same for therapy. For example, in imaging, if you won't use the same probe, if you want to do cardiac imaging or transrectal imaging for prostate. Same thing for therapy. We'll have different probe if you want to do a extracorporeal or intracavity application. Same thing for the frequencies. If you want to treat, look at the eye or look at the liver, we will have very different frequency range. And we'll do the same thing for therapy. Interestingly, also, we can play with the acoustic frequencies, just what is done for imaging, for example. You have Doppler frequencies or B mode, and we do the same. We can change the frequency if we want to perform thermal ablation or cavitation. So there is a real analogy in therapy and imaging. So back to this translational research, these are the two examples that I wanted to show you today with recent results. Note here on these two examples that they are the results of a collaboration between a laboratory, LabTo, an hospital. So the first project is with the Hospital of Lyon, and the second project is the Hospital of Paris, and company, so EDAP for the prostate and the Carrelation for the brain. So the first project is called Ablator. It's a long story now, but I will focus on the last results with the focal therapy. So yeah, say the long story. Keep in mind that the first patient was treated in Lyon for cancer treatment, was treated in Lyon with the very early prototype of the Ablator in '93. After that, many different prototypes were created. These are two examples. So the Ablator Maxis from 2000 to 2004. So this had two probes that were placed in the rectum of the patient for looking and treating the prostate. So first in imaging phase, as you can see here, and A more evolved version arrived on the market in 2005. It's integrated imaging. As you can see here, the high-flow transducer surrounded an imaging probe. So again, you had the imaging phase first, and then a therapy. The illustration of the frequency of treatment. Ablatherm is intended to be used for two applications. As a primary therapy, first for patients with cancer, older patients, not suitable for conventional surgery, and as a salvage therapy for different cases, like people who had surgery first, external beam radiation therapy, brachytherapy, or HIFU. Ablatherm is available pretty much everywhere in the world, except in Japan and in US. But there's currently a pending application, a 510K application in the US. So Sonocloud opened the door recently, so we hope that Ablatherm will go through in the near future. This slide describes the general principle of Ablatherm. So as I said before, we follow the endorectal route because it gives a good acoustic window to prostate. The treatment is performed under ultrasound guidance. As you can see here, the urologist will define where he wants to position the high-flow lesion, and the procedure will be completely computer controlled. So the machine, there are robots in the machine that will allow scanning the prostate and deliver elementary lesions inside the prostate. The procedure is performed under Russian anesthesia, and the exposure conditions are adjusted according to the intended use. For example, we change a bit the exposure conditions, whether it's a primary care patient or a patient who had already external beam radiation therapy. So the acoustic property of the prostate are a bit different. Up to now, the treatment quality was assessed using contrast-enhanced MRI in the days following the procedure. But you'll see that now things have changed. So as you can see on this example, half of the prostate was treated here and completely devascularized after the procedure. I mentioned before that this project started in the early 90s, and since then, things have changed in the management of prostate cancer. At that time, the patient had their cancer diagnosed quite late, and it was estimated that they lived with the cancer for about 7 years. So the objective then was to be efficient at all price. It was a single treatment approach. It could be like radical surgery or radical radiotherapy. But now, with progress in diagnosis, we can diagnose the cancer much earlier, and we want to avoid over-treatment. So patients can live for 15 years with prostate cancer. So the idea is to really, of course, control the disease, but also preserve the quality of life of the patient. So in that context, focal therapy was proposed, and HIFU is a really suitable option for this kind of treatment. So EDAP came up with a new version of the Ablatherm. It's called the focal one. This timeline explain a bit this translational process. The prototype was designed just after the release of when we started releasing the Ablatherm with the integrated imaging. So it was planned to start a new version of the Ablatherm. In 2009 and 2010, we performed preclinical trials while checking that the device was safe and as efficient as older version of the Ablatherm. Clinical trials started in 2011 with a CE mark in 2013. So what is different of this focal one compared to the older version of Ablatherm? It is based on 3 pillars. First, we want to localize prostate cancer, then we want to treat, and then we want to assess the quality of the treatment. So for localization, this version of ablatern offers the possibility to merge pre-treatment MR with perioperative ultrasound imaging. Before treatment, radiologists will define what are the suspect zone of cancer, that you can see on the image on the left. And once the patient is in the machine, this suspect zone will be displayed on the ultrasound images. So there is a system for co-registering the ultrasound image and the MRI image made before treatment. The treatment is also more precise because now the probe allows dynamic steering of the beam. The probe is made of 16 annular rings, allowing precise focusing at different depths inside the prostate. So here again, you can see the volume that the urologist decided to treat here, and the machine will position automatically the different focus that it should scan in order to treat this volume. So now with this dynamic focusing, it really allows to follow precisely a contour. And then it integrates the possibility to validate the quality of the treatment using, for example, contrast agents. It can be injected right after the treatment. You see that you can check that the zone which is vascularized corresponds to what you aimed before treatment. For example, here where you can see that all this part of the prostate was completely ablated. So up to now, the clinical strategy consisted in performing either a total ablation of the prostate or at least any ablation, as you can see here on this example, which was published last year, where half of the prostate was ablated and confirmed with MRI and gadolinium injection. Now we are we are going to our focal therapy. This is an example here where only one part of the prostate was had cancer, so we can limit the treatment to this part of the prostate. In the future, the goal is really to be able to detect the prostate cancer and limit the treatment to this zone, even if you have multiple multi-cancerous foci. In that context, the colleague in the lab has been working on a system for helping the diagnosis of localized prostate cancer. What he's doing is comparing different MRI modality with the histology that was performed on prostate that were removed by surgery. So this program helps calculate some kind of probability of presence of cancer. And this is an example here. So on the left image here, the program says in this case that there was 93% of chance of cancer in the first ROI and 98% in the other ROI. And in fact, on histology, it worked very well. So with cancers in both ROI. So that could be a good complementary tool for allowing the selection of the zone to be treated by urology. Interesting because it's very helpful in the detection of aggressive cancerous foci, Gleason score 6 or 7, and not very efficient for a lower Gleason score. So it's good because then in this context of overtreatment of prostate cancer, it's good that we really focus our attention on the aggressive cancer and not on the others. And it has been demonstrated that this kind of tool can be helpful for both non-experts and also expert uroradiologists. So in summary, for this prostate part, we could say that HIFU is an effective first-line treatment for localized prostate cancer. It preserves quality of life by presenting an attractive mobility profile. It can be repeated. Didn't show slides, but even if the treatment is not working, you can have other treatment like radiotherapy, for example. It's recognized and reimbursed in several countries, and it can be a salvage option for external beam radiation therapy failures. Due to the technology by itself, it can be an ideal modality for focal treatment, as you can really limit your treatment to one small part of the prostate if you are able to say where is the cancer. And I believe this is a good example of translational research with device for conformal treatment. But this is not the only one. Move to the second project, the Sonocloud. Sonocloud, so I said, is a collaboration between the laboratory, the Hospital of Paris at APHP, and Carrelation, which is a Paris-based company which aims at developing ultrasound device for neurosurgery. So with them, we have been working on an implantable ultrasound device for repeated opening of the blood-brain barrier and drug delivery into the brain. So the goal was to treat brain tumors, which we all know have a dramatic prognosis. Why is that? Is because the blood-brain barrier limits the efficacy of the penetration of the chemotherapy and by preventing the penetration in the tumors. So it has been demonstrated that ultrasound in combination with contrast agents can open this blood-brain barrier in many preclinical studies. Most of this study proposed to use in the future extracorporeal device, but the main problem is the skull. And that's an issue, particularly if you want to repeat the procedure in concomitance with chemotherapy. So you want a fast way to open the blood-brain barrier and an easy way for repeated application. So what we propose to do is to implant an ultrasound source directly into the brain just after surgery in the skull. So this is what we've been doing with using a small burr hole, a centimeter in diameter, as you can see here. And the idea is to position this device, as you can see here. And this device could be connected transdermally using a particular electrode here. So there is no internal power supply for a good MRI compatibility. So again, we use an extracorporeal power supply in order to generate ultrasound each time we want to open the blood-brain barrier. So the device is very simple. It's a flat piston, as you can see here, a centimeter in diameter, operating at 1 megahertz. The pressure range is between 0.5 and 0.8 megapascal, very similar to what others are doing with the extracorporeal device. Exposure duration is 2 minutes, and the PRF 1 Hertz with a pulse length of 25 milliseconds. So this is typically the pressure field that we can get, which is a simulated pressure field in water. But what we hope to do is to open the blood-brain barrier very quickly on a quite large volume around the zone that was resected. So typically, the syndicated zone corresponds to the dimension of the transducer, which is a centimeter in diameter, and the depth depends on the attenuation of the depth. So, of course, this project started much after Ablaterm because we designed this prototype after a request from Alexandre Carpentier from the APHP in Paris. This was done in 2010. We ran many preclinical trials from 2012 to 2014 on very different species, rabbits, dogs, and primates, in order to check the safety and the efficacy of the method. I will go through these results in the following slides. Then we've been to the regulatory approval process. It was very important for this implantable device to be strict in terms of sterilization, biocompatibility, and electrical safety, for example. And then in 2014, we started a phase 1/2 clinical trial with the first patient included in July 2014. So, as I said, concerning the preclinical trial, we started first with experiments in rabbits. just checking that with this very simple device, we could open the blood-brain barrier. So it's a very conventional experiment with event blue that penetrates very well in the sonicated part of the brain. So always we have contrast agent, low pressure ultrasound. We looked also on another model, dogs, where we looked at the toxicity after 7 days, well, feasibility of the opening of the blood-brain barrier, and also toxicity at 7 days. So you can see here on this MRI the penetration, enhanced penetration of gadolinium in front of the zone where the Sonocloud was implanted. So we didn't see any adverse effect on MR and histology, and no change, normal behavior of the animal. And at day 7, the MRI show a normal DVD. So these preclinical studies show the feasibility and the safety of the opening of the blood-brain barrier in the short term with single sonication and opening. That's a very important point because, as I said before, we will try to open this DVD at every chemotherapy session. So it's important after to look at the safety and the feasibility of a repeated opening of the blood-brain barrier. So we checked that we could enhance the penetration of different chemotherapy of different size. This was done in rabbits. Then the drug was injected 5 minutes before the opening of the blood-brain barrier. We tested the irinotecan and temozolomide. Obviously, the largest is the molecule, the more efficient is the procedure for increasing the penetration. We also tested with carboprotein, but I will come to this later. So on a more realistic model, we worked on the feasibility and safety of repeated openings of the blood-brain barrier. So this was done on primates. We had 3 baboons and 1 macaque. The protocol consisted in implanting the device on top of motor cortex, just like in a very conventional door hole, as you've seen before. And we performed the procedure every 2 weeks for 3 months. So it corresponds to 7 opening sessions. We follow up the animal with contrast-enhanced MRI, electrophysiology, PET, and histology. So on this slide, you can say that it's feasible to implant this device. So it was implanted here on this particular animal, and it's efficient. So it means that every 2 weeks we could reopen the blood-brain barrier. As you can see, the enhanced penetration of gadolinium always in front of the transducer. So and this was observed immediately after sonication. In terms of side effects, we didn't see any change in the cerebral metabolism with PET, neither inflammation on another kind of PET at 7 days after the application of ultrasound. The monitoring of the electrophysiologic monitoring of the animal didn't reveal any abnormality, and the 4 animals behaved completely normally for the 4 months of the experiment. On histology, at sacrifice, we didn't see any sign of hemorrhage or ischemia and HN staining. With all this preclinical package, we could start a phase 1/2 clinical trial. All these details, you can find it on clinicaltrial.gov, but I tried to make a summary. The primary outcome of this clinical phase was to evaluate the safety of transient restriction of the blood-brain barrier using this particular system. And the patient we were looking at had recurrent glioblastoma. In the course of these clinical trials, we wanted to look at other few things. First, you see the escalation dose for ultrasound. So we wanted to look at the maximum tolerated dose of ultrasound, starting from 0.5 megapascal up to 0.8 megapascal. We wanted to quantify the description of the DVD using dynamic T1 contrast-enhanced MRI, and looked at the progression-free survival in patients and also the overall survival in patients. The clinical methods follow. So we implant the Sonocloud. We increase the dose of ultrasound from the first patient to the last patient. It's a Simon design, so from 2 to 24 patients. We use carboplatin. We checked, we didn't show it, but we checked that the system is also efficient for the penetration of each drug. So we had 6 cycles of carboplatin every month, and the dose of ultrasound of carboplatin is decided according to renal function and IUC. The main inclusion criteria, there is a very long list on the clinicaltrial.gov website, but if you summarize, we wanted to treat patients with recurrent glioma who have failed standard therapy with surgery and or treatment with radiation and temozolomide. The main exclusion criteria relate to the use of the different method of imaging we are using for following our treatment. So there should be no contraindication to MRI or gadolinium or contraindication to echographic contrast agent, because this is what we are using for opening the blood-brain barrier, obviously. So these are the early clinical results. We treated so far 15 patients, which corresponds to 32 procedures. What we have seen, it's perfectly possible to implant the Sonocloud in every 15 patients, as you can see here. This is the very first patient who had surgery. This part of the nerve was removed. And Alexandre Carpentier wanted to position the device here in order to sonicate the periphery of the zone infected. And on the last patient with the highest dose of highest pressure, we could evidence the first opening of the blood-brain barrier, as you can see here, with the increase in the penetration of gadolinium just in front where the transducer was put. So, in summary for this Sonocloud study, we designed an implantable device for opening the DBD post-surgery. It's called Sonocloud. It is feasible and safe and can be repeated in preclinical models. We've seen that it improves the drug uptake in syndicated volume in preclinical models again. And the early clinical results demonstrate the feasibility, the efficacy, and the safety of the concept. So overall, what I'd like to leave you as a take-home message is, for us, it's very important to start from the clinical unmet need first. And the technology is just an enabler. We will design particular device for each medical problem. And it's very important, in my opinion, and in the opinion of my colleague, to involve different actors of the project, even at the early stage, taking into account their clinician and industrial point of view. So, to conclude, I'd like to thank a few persons. Jean-Luc Chaplon, director of the lab, a colleague, François Chavrier and Olivier Roubière, a colleague from Hospital Civil of Lyon, Albert Jolet, who performed the very first treatment of prostate cancer with ablator, and his colleague from IDAP, Emmanuel Blanc. Same thing for people at Cartera, Alexandre Carpentier and Michael Canning, and also the group of Lacto, you can see on the picture below. Thank you very much for your attention.
So I was wondering, just using these examples, what did you find to be the sort of biggest challenge in bringing something from the kind of preclinical stage into actually developing it into a product?
Biggest challenge? No. Maybe the fact that it's a very long process. Particularly, it takes a very long time to make sure that your treatment is efficient. So you see this prostate project started in 93 or something, and still now we are struggling to get approval for them in the US market. So that's a real challenge. I don't know how long it will take for the other project in the brain. but that's a real issue. We think always that the technical part is very complex, but no, it's more like the clinical important.
Thomas Kusek from Electa, been involved in the development of a number of therapeutic devices, said that for every dollar that they spend on technical research and development, they have to spend $1.50 on commercialization.
I can only agree with that. It's a long process.
So I had a couple questions about the Sonocloud device. What are the use cases for a device like that that's implantable as opposed to an external transducer that's designed to penetrate? And then also a question to go along with that. When they were looking at the chemotherapy agents, were they using things that were also activated by the ultrasound or released in just that area, or was it systemic chemotherapy that then the blood-brain barrier opening allowed to penetrate?
I'll start that second question. So it was like really conventional chemotherapy that was injected systemically. So the idea was really to try to be combined with the conventional chemotherapy process. Okay. For the first question, the objective of developing this implantable approach was to facilitate the clinical transfer. Each time, because in this case, it was really important that the procedure could be performed in parallel to each chemotherapy session. And this comes very often, so it has to be fast. You know? And this is why very early we decided to not go through the cell and other system that you just have to plug and play. Very easy. So that was the reason why we did that. And anytime we would have to repeat the procedure, then this approach is interesting.
So I have a related question. You said that the device is plug and play, and you showed that it's powered by an external source, so that the device itself is MR compatible. Maybe you can't go into details, but I'm curious to know the power coupling between that external device and the transducer. Is that inductive, so through the skin, or is it a physical connection that pierces the skin?
No, it's really physical connection, as you can see on this image here. So only this part of the Sonocloud is implanted in the skull, just like that. And once the patient comes back to the hospital for the surgery, for the chemotherapy, you have this needle that will go through the scalp and will be connected straight into the Sonocloud. So it's a transdermal connection. There is a wire that goes to the amplifier and the transducer.
Mine. How do new projects come to you, and how do you choose what to work on next?
That's a good question. The new project usually comes from doctors. As I said, it's when they have, well, they know of iFood. I heard about the technology. They have a problem that they think ultrasound can do something for it. And for example, this project with Alexandre Carpentier is a very good example. He came to us because we had this idea of implanted device. So I think for the first question, it's really the clinicians who are bringing the project. After we have to find a solution, which is not always. Sometimes we can't. How do we choose what to work on next? Well, it's a combination of different things. First, sometimes it's not feasible. I mean, people are coming with an idea and saying, well, ultrasound cannot do anything with that. There is, the challenge. Is it difficult? Yes or no? Does anybody else do something related? So we want replicate an existing research. Funding is also important. If it's like an application with a big market, then it's easier to work in that direction instead of trying to solve a very rare case. So it's really a combination of different things.
And that concludes today's webinar. Thank you all for joining us today, and please keep an eye out for an announcement for our next webinar.